Okay, so my topic is a little bit different from what has been presented so far, but I'll try to make it quite simple and brief. So I will talk about natural hypothetic systems as a new cryoprotected agent. So the first question that you might be asking yourselves is that what are natural hypothetic systems? So basically, they are homogeneous mixtures of substance that have a melting point far below the melting point of each individual component of the mixture. So this mixture comprises the nitrogen bond donor and a nitrogen bond acceptor, and this mixture is mainly formed through intermolecular hydrogen bonds. The most common mixture that we found in literature is the choline chloride urea. So basically you have two solids, then you mix them, you stir them, you heat them, and after some time it becomes an homogeneous, transparent, viscous liquid. What are the main advantages of these new materials that have emerged in the last decade? So they are biodegradable, they have presenting low cytotoxicities, there is no waste during their production because you just have to mix the components. They are quite cheap to produce because you use cheap raw materials, you don't have purification or byproducts because you don't have a chemical reaction. When these components are natural molecules like sugar alcohols, and even water, we call them natural hypothetic systems. The most common example is honey. All of us have honey in our kitchen. Honey is nothing else but a mixture of sugars that become liquid. Sometimes it becomes more solid during the winter time because of cold, but honey is the most common natural hypothetic system that you can find. So in our group, we have studied the thermal properties of the systems. We took just one example, glucose proline, in this molar ratio, 5:3. We found out that these systems are glass formers, so they do not crystallize. And these systems have an impact on the crystallization and vitrification of water, as you can see here in these images. So when we have only water, you have the melting and the crystallization temperatures well defined. But once you start adding these systems, you start to see a shifting in these temperatures. And when you have about 80% of our systems with the 20% of water, you do not see crystallization or melting. So this was a work that was was inspired by nature, because some animals that survive extreme temperatures, climates, they are known to accumulate certain metabolites in their body. In some cases, the type and the amount of these metabolites depend on the season, and these components that are mainly sugars, polyols, amino acids, as I showed before, are known to form natural hypothetic systems. So what can be the role of these systems in cryopreservation? So basically, as all of us know, in cryopreservation, the cell viability is highly compromised by the formation of ice. And normally we use a cryoprotected agent, generally DMSO or glycerol, but they are highly toxic for the cells. So we started to think, what if we replicate what animals and plants do to survive the cold environment? Can we translate this process to the cryopreservation and the cold storage of biological materials? So I'll be presenting two works that we have been doing in our research group for the past 2.5-3 years. So in this work, we prepared a huge library of natural hypothetic systems. They are composed by sugars, polyols, and amino acids, basically, and water. Some of them have water, some of them don't have water. They have different combinations, they have different proportions between them, the molar ratios. So the first step was to study the cytotoxicity of the systems. As you can see, they are non-toxic. Most of them present a high C50 values around 2 molar range, so it's quite high. And then we studied the viability at 5, 10, and 20%, because as many of us know, DMSO is added to the cells at 10%, so we wanted to see what happens with our systems. So as you can see, at 20%, cells do not tolerate this concentration. At 10%, most of them present higher than 50% of cell viability after 24 hours of incubation. And with 5%, our systems presented pretty much 100% of viability, but if you compare with DMSO, it's only 50% of viability. So this is quite promising results at that time. So then we moved on to the freezing process. So we froze 2 cell lines, L929 cells and ACAT cells. They are quite normal cell models. So basically what we did, we froze the cell lines with our systems at 10%. We used 10% and compared with DMSO at 10% as well. And after more or less 2 weeks with all the cells, and we measured the viability of the cells at 24, 48, and 72 hours. So as you can see, for the L929 cells, our systems worked fairly well. In a similar way as DMSO, but for ACAT cells, the systems worked pretty much better when compared with DMSO. One thing that needs to be highlighted is that we did not remove the CPA after the thawing. So basically, the cryoprotected agent, either DMSO or the Eutectic systems, remained in the culture during these periods, the 24 up to 72 hours. So to mimic a process that if you want to use the cells without washing, without removing the freezing media, you can do it with our systems. So after that, we chose our best systems, and we studied the influence in the cell viability, in the cell membrane, in the proliferation. As you can see in these images, after 72 hours, you can see that cells proliferate pretty well. These are the L929 cells. You have pretty much good colonies of cells. The cell membrane was normal. So this showed that these systems can be used. So for the ACAT cells that we got pretty much almost the double of cell viability with our systems. You can see beautiful, huge colonies after 72 hours with our systems. Sorry. So after that, we thought, okay, so what about freezing the cell lines in -20? Because not all of us can afford to have a cryotank or -80 freezer. So we did the same study for 2 weeks in the cells. So we froze the cells with our best systems. We froze them at -20. As you can see, worked pretty well. Cells viability were far good as DMSO. In a similar way for the ACAT cells, they are quite sensitive cell line. Cells recover pretty well after the thawing. So these were really nice results that we got at the time. We published the results, but I'll show you this new report that we just published last week in Scientific Reports. So basically, we wanted to see if we can use this as a new vitrification media. So in this case, it was a very similar study. So I just wanted to say that the difference between slow freezing and vitrification, the main differences are the speed of freezing, but also the concentration of the CPA. Normally, the slow freezing process, like the one that I just showed, you normally use slow amounts of CPA. The process is simple. The speed of freezing is normally slow. That's why it's called slow freezing. However, for the vitrification, we usually have very high speeds of freezing, and also the concentration of the CPA is very high. So in our study, we studied our systems, that library that we prepared, in 50% of concentration instead of 10%, and then we froze the cells. So basically, it's the same procedure as you do with the normal cells. So you take the cryo vial, you put the cells, you put the CPA, but instead of putting the cryo vials in the Mr. Frosty and then putting in the -80, in here we just immersed the cryo vials in liquid nitrogen, so that the samples are completely frozen after a couple of seconds to mimic the vitrification. So we did that, and after 2 weeks of freezing, we thawed the cells, and then we measured the viabilities that I'm showing here, and we measured it after 24 and 72 hours. And you can see in the L929 cells, they recovered pretty much well. They did good. For the ACAT cells, only one system showed promising results, but we were not discouraged by these results regarding the ACAT cells. So we saw again the cell proliferation. We analyzed also the cell membranes, and we observed that the cell membranes were quite normal. The cells proliferated pretty well after 72 hours. As you can see, the cells are alive in this live death assay. And for the ACAT cells, the only system that showed promising results. So as you can see in this picture, after 72 hours, you have a completely confluent cell colony. So this showed that these systems can actually be used as a vitrification media. So this opens the opportunity for our systems, the natural dipathetic systems, to be used for other type of applications. We are starting now to study in some stem cell lines. Also, it could also be good for egg and sperm vitrification as well. So these were quite promising results and very interesting. So just final remarks. So we prepared these new natural dipathetic systems that are composed by metabolites, normally used in animals that can tolerate the cold environments. These systems are liquid at room temperature, and they are glass formers. The biological performance were studied in these presentations, as I showed you, and were compared with the DMSO, which is the golden CPA. All of them showed low toxicity. L929 cells showed similar results to DMSO. However, for the ACAT cells, we showed an improvement in post-thawing cell viability. And we also observed normal morphology and normal proliferation in cells frozen with the natural dipathetic systems, even at 50%, even in the vitrification studies, we saw that. And we also observed that these systems can improve the ability to freeze cells at -20 instead of using cryotanks or -80 freezers, only up to 2 weeks. We know now that cells don't tolerate 1 year, for example, with these systems, but we can freeze for up to 2 weeks in the freezer. So sometimes these can be of use in some studies, in some cases. So basically, just to say that our results show that these systems, the natural dipathetic systems, can actually be used for other type of applications in the cryobiology field. So I just want to thank the funding agencies to support this work, my team, the Deshoff Group and the Biocatalysis and Bioenergy Group at Nova in Lisbon, Portugal. So I'm quite pleased to answer some questions you may have, and thank you so much for your attention.
Thank you so much for such an interesting talk with lots of impressive data, because now I can imagine that we will get plenty of questions, and we have time only just for a few of them, but still I hope that we will find some time and we will try to just prolong a little bit probably the session, because yeah, it's like very impressive and we want to ask you a few questions. So the first question is from Natalia.
Thank you for this very interesting report. I am glad to meet you. I am familiar with some of your works, in particular the articles in cryobiology last year. I think it's a very comprehensive study. I have some practical questions. The first, how stable are these natural deep eutectic system needs? Their storage condition.
So you can store them in the bench at room temperature. Some of them are quite stable. Some of them crystallize after some time, but you can heat them again. You can stir them and heat, and then you recover the system again. So they are quite stable, I can say.
Thanks. The second question is it always necessary to control the process of preparing nails using certain methods? Or you can check once and then prepare these systems according to the protocol without any methods?
I did not understand, sorry.
Yes. Лен, help me. Надо ли каждый раз проверять приготовление этих систем? Или можно один раз проверить и потом по протоколу готовить?
Do you need to check each time the protocol for preparation of the system? So like, or you can create the stock of these cryoprotectants, mixtures and use them, or do you need to check each time their quality? Do you need to create the new mixtures each time, or you can use once prepared for quite a long time?
Yes, so you can prepare a large amount, like 20 milliliters or 50 milliliters, and then you can use it once you need. You don't have to prepare it each time.
So there is no need in quality control. So it's perfectly stable and they work.
Yes, yes.
Yeah, because it's inspired by nature, and everything that is natural, it's usually good.
Yes.
The last question is, what can you say about osmolality of this system? I can explain my question. I work with erythrocytes, which are very sensitive to change in osmotic pressure. And I want to understand, can I use these systems, these needs for my research?
Use these systems with the
erythrocytes. Erythrocytes are quite sensitive to osmotic changes. That's why it's like, and here you got high concentration of different sugars, and it means like high changes in osmotic conditions. So that's why there is a question: can your mixtures, your solutions, be used for erythrocytes, for example?
At this point, we don't know. We only studied these mammalian cells. So this is like the next step is move on to other organisms and the other cell lines.
Thanks.
Thank you so much.